Hello students, today we are going to discuss in detail on gene therapy. One of the challenging ongoing research in the genetics. Still it is promising. The gene therapy is the method to treat various genetic disorders because the mistake lies in the DNA. It's at our first job where we can go and rectify the mistakes which is present in the case of our gene. The scientists are using to treat inherited genetic disorders like sickle cell anemia, thalassemia, all these genetic disorders. They are trying to treat with the help of our gene therapy. Various different monogenic and polygenic disorders are evolving every day due to accumulation of mutation. It is forceful and it is made out of exposure to the environmental radiation like alpha, beta, gamma radiation as well as ultraviolet radiation. Along with that, on exposure to various environmental mutating agents that are responsible for the development of changes in the genetic gene level that resulting in various genetic disorder. It may be monogenic means single gene or more than one means polygenic gene disorders. In gene therapy, the normal functional protein is revived by replacing a mutant gene. Normally, when a gene is undergoing mutation, it will not produce the normal protein because there will be a defect in the case of coding region of mRNA that lead to change in the sequence of amino acid in the protein, or the protein may be truncated by the arisal of stop codons, or different amino acids will be present. That is, the amino acids like acidic amino acids may be replaced by basic amino acids that alters the property of a particular protein, resulting in a problem. It's a very challenging part of this, where we are going to discuss in detail about the gene therapy and its various type in this particular video. Actually, it was evolved in 1989. The first patient was recovered with the gene transfer technique. So we are going to discuss in detail with example. So what are all the key topics that we are going to see, and history of gene therapy that you can read from the textbooks. And what is gene therapy, we are going to deal in detail, and types of gene therapy and different techniques of gene therapy, that is vectors for gene therapy, viral and non-viral vectors, and how the stepwise process of gene therapy is carried out, what are all the applications of gene therapy, limitations and challenges of future aspects. And here on the right side of the table, which shows some of the abbreviations which are relevant to our gene therapy. So, what is gene therapy? We are going to introduce a new gene that is replacing the mutated gene. That is, the faulty genes are replaced by correct genes by this mechanism. That is what you call it as our gene therapy. So, by definition, we can see the process to recover the normal function of the gene by transferring a correct gene to that place or removing the faulty gene is known as gene therapy. There are four different types of gene therapies are most popular worldwide, that include somatic cell gene therapy, germline gene therapy, in vivo gene therapy, and ex vivo gene therapy. Somatic cell gene therapy means that involves only the body cells where the mutations are rectified in the body cells, whereas in the germline gene therapy means in the germinal tissues like ovary as well as in the case of the testis, where the corrections are made in the gene level so that at the initial stage of the fetus development itself we are trying to rectify this and to solve this issue with the help of our gene therapy. Another one is in vivo gene therapy, means we inject our correct gene internal to the inside the body of a particular organ where the defect lies with the help of various viral vectors, that is called as in vivo gene therapy. And ex vivo viral as well as non-viral vectors are utilized for the in vivo gene therapy. Whereas in ex vivo gene therapy, what we do is we take out the body cells and we will add the correct gene and we will put it back to our body for the treatment procedure, that is called as ex vivo gene therapy. So further, the somatic and germ cell the gene therapy are categorized based on the cell type that are involved, whereas in vivo and ex vivo gene therapies are categorized based upon their application. First and foremost, somatic cell gene therapy. The cells other than germline cells are called somatic cells, like bone, blood, skin, internal organs, and other various vital tissues developed from the somatic cells. Normally, they undergo the process of mitosis. Those cells which are involving mitosis are coming under the somatic cells. Here, 46 number of chromosomes with 23 pairs are present in the somatic cells. In the somatic cell gene therapy, what we do is a mutant gene will be present in the somatic cells or in turn replaced by the transgene or correct gene. So normally the change what we make remain lifetime only. It will not be transmitted from one generation to the next generation. Normally the genetic disorders are inherited from or passed from one generation to the next generation. Unless we treat in the germline level, it cannot be passed to the next generation. So here in the somatic gel therapy, whomever the patient where whom we are treating, the problem will be solved. That if the problem or if the disorder continues with the next generation of the daughters or their granddaughters or grandson or the son, that time the defect will be present in them due to passage of the defective gene uh, by hierarchy. Normally, the somatic cells are not involved in the reproduction, hence they can be inherited. Various ethical issues are involved in the somatic cell gene therapy, but the success rate is noticeable in the case of our somatic cell gene therapy. Normally, the virus-mediated gene transfer and liposome-mediated gene transfer are widely used in somatic cell gene therapy. The common target is bone marrow cells. Cells which are present the bone marrow cells that can be divided throughout the lifetime, and they produce different types of blood cells. So we target the somatic cell gene therapy mainly towards our bone marrow for the process. In this process, the virus like adeno-associated virus is commonly used as a vector for the somatic gene therapy. It infects isolated bone marrow cells. Once the gene gets inserted into the target cell, that is the virus AAV, adeno-associated virus. Within the virus, the unwanted regions of the virus genes are in turn replaced with our gene of interest that contains the correct gene without any mistake. Then what we do is we'll allow the virus to infect the isolated bone marrow cells in vitro in the laboratory. Then once the gene is transferred to the bone marrow cells, what we do is we will take it up and we will put back to the bone marrow itself. So this is the broader view of the entire mechanism of somatic cell gene therapy is shown in the figure. The somatic cell gene therapy has several limitations. The virus that combines with the host genome and infects the cells. There is a virus escape also possible when we allow it to enter inside. And if the gene does not insert to the specific location, then automatically it disrupts the other normal gene also. The chances of activation of oncogene and proto-oncogene are also very high in the case of our using AAV, that is adeno-associated virus gene therapy. So, what is oncogene and proto-oncogene? So, when the some of the viruses which are capable of causing cancer, when they infect, the gene of these virus get inserted into our body, but as sometimes it is not expressed. When it gets activated only, it is converted from the silent form, that is the proto-oncogene, to the oncogene form. There it starts developing the production of cancer. So if we allow this treatment procedure of somatic cell gene therapy along with the bone marrow cells, the AA virus also get inserted, Then what it is doing is by mistake which activates a proto-oncogene to a oncogene. That time the problem occurs in the case of a system. And moreover, it alters only the actively dividing cells where it integrates into the nuclear chromosome. That is the nature of the adeno associated virus. Next one is germline gene therapy. So before splitting the embryo, what we do is we take out the embryo containing germ cells. It may be sperm or egg cells. The fertilized egg or embryo. This method is to which the gene is transferred is called as germline gene therapy. Consequently, the changes are made in the DNA. These cells can pass to the next generation also. But this is we are playing as if we are playing with the life of the embryo. Some scientists believe it is unethical. The germline gene therapy is prohibited worldwide because of this, as if we are playing against the god. We are taking out the embryo and we are adding some gene. There is more possibility that we can alter the function of the embryo also by this method if we are not going to have a safe play as by the ethical guidelines. So it will lead to other complications also in the case of the fetus production. So that is why it is considered as against the willing of god. However, the comprehensive view of this method is shown in the diagram. Using the viral vectors, the cells of the reproductive tissue or unsplit embryos are infected. The modified cells are grown in the sterile laboratory as if we are doing the IVF, in vitro fertilization. Same manner, what we are doing is we are taking out the embryo and we are adding the gene by using an virus and we infect it. Then the modified embryo cells are then taken out and the unmodified cells are removed. Then these modified cells are then allowed to fertilize in vitro then by artificial reproductive technology, that is IVF. Then the gene containing the embryos are further transferred to the uterus of the mother for the development of the fetus. This is what shown in the figure. The next one is in vivo gene therapy. In vivo means the exogenous gene is directly injected into the target cell so that the gene of interest is introduced into our body by aerosol, by spray or by the injection. So automatically, directly the gene can enter into the cell, but the thing is that it cannot spread from the one cell to the other cell. Here what we are doing is, as per the guidelines, the ideal example of in vivo gene therapy is used for cystic fibrosis and Duchenne muscular dystrophy, in which the protein dystrophin is defective in nature. So the kids will have complaints related to muscular disorder that cannot be treated, and as well as in the case of the cystic fibrosis that develops in the case of the lung, that alters the functioning of the lung. So in cystic fibrosis, the exogenous gene, that is the transgene, is introduced to the nasal spray or aerosol, whereas in the case of the Duchenne muscular dystrophy, a dystrophin gene is inserted in the muscle through the injection. So some of the surrounding cells are infected without any reason and causes some adverse effect. But conclusion, as we conclude that in vivo gene therapy is not a good option. Ex vivo gene therapy. In ex vivo gene therapy, the affected cells are collected and they are modified in the laboratory outside the body and they are inserted back to the body. Here none of the gene therapy poses are performed inside the body, hence it is called as ex vivo. So we can control the whole process in the laboratory and we can check once and twice so that there is no any other associated viral infection before it gain entrance, as well as there is no alteration in the case of the embryo level also during this ex vivo gene therapy process. Here the bone marrow and blood cells are often utilized for the gene therapy process. We know that it is actively dividing cells and it is involving the adeno associated virus. But cystic fibrosis and Duchenne muscular dystrophy cannot be treated by this method. It is not successful by this ex vivo gene therapy mechanism. Why we are to choose actively dividing cells? Normally, the transgene therapy is involved using done with the help of our actively dividing cells. Therefore, the blood cells and bone marrows are also best choice because they are always undergoing division. They are pluripotent in nature, so they are dividing actively till our lifetime. So we are choosing those cells for the purpose of it. And many of the blood related disorders can be treated like thalassemia, sickle cell anemia, thrombosis, hemophilia can be treated very effectively by this method. The defective or mutant cells are isolated from the patients and the exogenous gene of interest, that is good gene, is inserted to the cell lines using the viral vector under sterile strict conditions. The altered cells are grown, the modified cells are taken and purified, and we remove the infectious virus and we will check it that is of in good condition, aseptic condition. Then these modified cells are injected back to the body of the patient. So, widely used therapy where we make use of the vectors for the transfer of gene involving a retrovirus as well as adeno associated virus. In the case of the retrovirus, the epsilon sequences are first removed so that the virus can insert their own gene into the host genome. So there are different techniques of gene therapy. The loss of function mutation and abnormal gene expression are the two key reasons which causes genetic disorder. And based on this technique, there are three types of gene therapies are there: gene augmentation therapy, gene inhibition therapy, and suicidal gene therapy. Gene augmentation. The natural function of the gene is lost due to mutation, and a loss of function in the mutation that is taking place. What we do is the normal protein cannot be translated. In augmentation process, what we do is we are replacing a fully functional wild type, that is the normal type of the gene that produces into a correct protein without any defect by this disorder. This is the majorly used monogenic disorder treatment procedure that is done. Especially the cystic fibrosis like disorders can be treated using the gene augmentation therapy and gene inhibition therapy. Gene inhibition, overexpression of the particular gene that is threatening to the lifetime of a person, for example like cancer. Such cases, the changes in the methylation pattern or epigenetic profile of the particular gene can lead to overexpression or underexpression in the case of the uh, person. Such case we go for gene inhibition therapy. In the gene inhibition therapy, the overexpressed gene is inhibited by using another gene or DNA sequence, or we make use of interfering with the activity of the product that can able to suppress the overexpression of the particular gene, which is responsible for the development of cancer in this case. This is the present gene therapy is best choice to treat inherited disease, infectious disease and cancer. The overexpression oncogenes can be lessened by using this method. The next one is suicidal gene therapy. It is especially useful and necessary to kill the cells which are associated with cancer. In the case of suicidal gene therapy, it is very necessary to kill the cancerous cell because it is uncontrolled cell division that takes place. Such cases, the suicidal genes are introduced to kill the cells. The gene products are toxic products that are the protein that induces the strong immune response against the cell that leads to the cell death. Such suicidal gene therapy is specially designed to cure the cancer. A brief view of what these are explained in the diagram. That is our in vivo, ex vivo as well as suicide. That is, yeah, sorry, gene augmentation, gene inhibition, and suicidal gene therapy is shown in the diagram. We'll see the rest of the content in the next video. Thank you.